Okay, so hi everyone and welcome to BrainMap. It's my great pleasure to introduce our speaker today, Dr. Livia Tomova. She is interested in how stress, loneliness, and social isolation affect the brain and mind, especially during adolescence. Her PhD research at the University of Vienna with Klaus Lamb was completed in 2016 and focused on the effect of acute stress on social cognition and the underlying brain processes. Her dissertation received the Austrian Prize of Excellence for Best Austrian Dissertation in 2016. Dr. Tomova then completed 3 years of postdoctoral training in cognitive neuroscience at MIT with Rebecca Sachs, investigating how deprivation of social needs affects the human brain. Dr. Tomova was awarded a Henslow College Research Fellowship at Hugg's Hall, starting in fall 2020, during which she aims to identify brain markers predicting vulnerability to the adverse effects of isolation and loneliness in adolescent and young adults. She's also interested in investigating whether and how social media can fulfill social needs. I would just like to remind our audience to please address any question you have, even during the talk, using the Q&A box. Dr. Tomova, thank you so much for coming here today. The virtual stage is all yours.
Thank you. Thanks so much. Thanks for having me. So today I will be speaking about fMRI signals of craving following acute social isolation in the human brain. And this is research mostly from my postdoc at MIT, but towards the end, I will speak a bit about my current research at Cambridge and what I'm planning to do. And as I was saying before, please, if you have any questions also during the talk, feel free to interrupt me. I'm happy to answer them. All right, so I'll start by giving a definition of what I mean when I say social craving. So theories in social psychology have proposed that there is a need to belong, which is a fundamental human need, and that loneliness acts as a signal, just like hunger or thirst, to signal to an individual that something is lacking and that it needs to take action to repair what is lacking. And indeed, studies on brain processing have shown that social cues engage the brain reward system just like other non-social rewards. And this has been shown in human studies and also in studies in animal models in social animals. So this suggests that social interactions in and of themselves seem to be rewarding. However, the sort of other side of the coin, so how the lack of social connection is represented in the brain, has not been investigated so much. So the question is, would that be similar to other forms of craving, like food craving, or would other brain regions represent a craving for social contact? So this can inform us about how social motivation is represented in a healthy human brain, which then can inform other research that studies these processes in clinical samples. And we know that many mental disorders affect social motivation, so most prominently autism spectrum disorder, but also depression and anxiety. So understanding better how social motivation is represented in the brain can inform these studies in patient populations. So the overall logic, how we sort of tried to approach this, this study was, first of all, that we started with this hypothesis that we know from other studies on motivated behavior that the deprivation of a need will result in neural signals of craving, which will then ultimately trigger a behavioral response and motivate the individual to do something about this. So the question is, would isolation trigger the same thing? And indeed, there are suggestions from previous studies that this might be the case. So our colleagues were looking, who then ended up actually collaborating with us in our project, but they were firstly doing the study in a mouse model. And here they were studying how isolation of mice for 24 hours was affecting certain neurons, which were dopaminergic, in the midbrain and their activity. And specifically, they were focusing on the dorsal raphe nucleus. So as you can see here in this graphical abstract, oh, I don't think you can see my mouse, or trying to, oh, sorry. Well, anyway, what you see is that these in the study, they found that activation in these neurons was changed after isolation, and specifically whenever the mouse was reintroduced to social contact, these neurons would start to show increased activation. And then they used optogenetics to study how inhibiting or activating these neurons would affect the social behavior of the mice. And what they found was that photoactivation of these neurons resulted in an increase in sociability. And what they also showed was that the signal was not representing a reward response. So they used a paradigm called condition place preference, where they tested whether the animals would start to prefer places whenever stimulation occurred. And actually what they saw was the opposite. The animals started to avoid these places. So that suggests that the signal was an aversive one. And we concluded from this that it was sort of like a loneliness-like state that created this drive to re-socialize. And what they also found was that inhibiting these neurons would decrease sociability, suggesting that they play a crucial role for our social motivation. So this suggests that isolation leads to affects dopaminergic neurons in the midbrain. And indeed, this is in line with this general model that deprivation creates neural signals of craving, which ultimately create a need or a drive to change one's behavior. So this was shown in the mouse model, and our question then was, well, is this similar in the human brain? So is there a homology in these processes between rodents and humans? So our plan was to study in humans how acute isolation would affect signals of dopaminergic neurons in the midbrain. So in order to do the study, we kind of faced several challenges, but the first one that we had as a major challenge was, well, would we be able to evoke acute social isolation in people? So people are not exactly the same as rodents in many different ways, but one thing that people have is that we have sort of this mental representation of other people. So what we know from studies on loneliness is that people are not necessarily feeling alone if they're objectively isolated, but they can feel alone even in a crowd, while also not feeling alone when they are alone, because they have this idea that others are still sort of in their circle and things like that. So it was not clear to us how we would be able to really induce an experimental setting where people would feel isolated. So what we did in order to test that was that we asked people who were pre-screened. So we screened them for social connection to only invite those who did not already experience loneliness or isolation in order to not conflate chronic and acute isolation. And then we asked them to undergo 10 hours in a room without any social contact. And the way we did it was that they did not only have no real life social interactions, but also no virtual social interactions. So they came into the lab at BCS at MIT, and then they spent there in one room 10 hours and were not able to access the internet or call their friends or things like that. We also asked them to refrain from engaging in like fictional social interactions, meaning that they should not read books or watch movies where they would have maybe their favorite characters, which also is known in humans to create a sense of connection. So we asked them really to spend these 10 hours in a state of a very reduced social, very reduced social stimuli, or actually none. And then what we did was then we scanned them after these 10 hours. And in order to get them into the scanner, we kind of came up with this procedure so that we would not have to interact with people after the isolation. So the way we did it was that we initially, before people started isolation, we trained them how to go into the scanner on their own. And then when they, after isolation, did that, we only had one research assistant watching them and making sure everything was okay, who was wearing sort of a full scrubs and a face mask to also avoid non-verbal social interactions. And what we also did during this isolation period was that we asked people throughout isolation how they experienced subjectively being isolated and how lonely they felt and how much they craved social contact. So this was kind of as a validation on whether our experimental manipulation worked or not. And what we found was that indeed people who spent these 10 hours in isolation showed on average an almost linear increase in feelings of social craving and loneliness as a function of duration of isolation. So you can see this here in the violin plots. So plotted is the mean rating on how they either craved social contact or loneliness at each 2 hours. So this suggests that we were actually able to induce a subjective experience of isolation and loneliness in our participants. So our second sort of major challenge in this project was, well, would we be able to measure the relevant signals that we are interested in using fMRI? So we were planning to do the study as an fMRI study. However, fMRI has some disadvantages in if you want to look at a specific neurotransmitter system, which you obviously cannot do that easily. So we were not quite sure if we would be able to translate the study that was done in animals, where they did direct recordings from single neurons using fMRI. So the way we did it was that we used food craving as a sort of proof of concept method, where we asked each participant to also undergo food craving. And then we would try to validate or find methods where we would be able to measure craving signals in response to fasting, and then compare those to those that we would find after isolation. So the way we did it in the end was that we asked 40 participants to come into our lab. We screened them, as I said before, and we also screened them for eating behavior. So as food fasting was our control condition, so we made sure that they did not have eating disorders or were currently on a diet. And then we asked them to undergo 10 hours of food fasting. So here participants were not isolated, so they also did not come into our lab. We asked them to undergo their days as they would normally do and just refrain from eating. But we also asked them to fill out an online questionnaire every 2 hours, so to tell us how they experienced food fasting, so we would be able to compare this to the subjective experience of isolation. And then we also asked the same people on another day to come into our lab and actually undergo the isolation. And we also included a baseline session where participants were not undergoing any form of deprivation, but just came into our lab and were scanned to make sure that in case we would see interactions between fasting and isolation, we would be able to quantify them somehow. And the order of these scans was counterbalanced. So just to show you also the subjective ratings for food fasting, you can see that participants here responded quite strongly. So they showed increasing craving of food and hunger, again, as a function of duration of fasting. And this was quite strong, which was expected. So now I'll just walk you through our plan, how we analyze the food fasting data and sort of validated that our research approach was working, and then I'll speak about the actual results in the isolation session. So first of all, I'll talk about the regions of interest that we selected, the challenges that we had for our functional scans, then how we actually measured craving, and our different forms of data analysis. So we did a univariate analysis. We also looked at correlations with behavioral measures and multivariate analyses. So to start with regions of interest, so one thing that we kind of were thinking about for a long time was how to, which regions to measure in the human brain, starting from the results that were found in the mouse brain. And one thing is that the function and the anatomy of the dopamine complex does not directly correspond between rodents and primates. So in primates, aversive motivation is primarily coded in the substantia nigra. So you can see this here. Yeah, I can't point to it again, sorry. But yeah, it's labeled as SN. And kind of one of the advantages of the substantia nigra is that almost all the neurons within this brain region are dopaminergic. And there are studies showing that fMRI signals collected from this brain region are quite likely representing dopaminergic activity. So this has been shown in studies that use both PET and fMRI in a combined way that showed that signals that are that people record with fMRI correspond quite closely to dopaminergic activity. So our target ROI was the substantia nigra, and we also included the ventral tegmental area because the anatomical borders between those two are not very strong in primates as opposed to rodents. And we also included the striatum as its major target of projections from the SNVTA, and it has been shown in many studies on drug craving and food craving that signals from the striatum also sort of underlie experiences of craving. So the way we then scanned this brain region was that we had to, again, face some challenges with this. So first of all, the midbrain has a lot of small structures, and in general, there is quite a strong difficulty in localization of these structures. And also there is a sinus right in front of it, which causes distortions and signal loss. So I'm showing this here, and you can see the midbrain and the sinus in front of it. So the way we did it then was that we used a method that has been recommended in the literature, which is to tilt the slices upwards, the ACPC line, so then to make sure that the slices that would be the ones that collect data from the midbrain would not also go through the sinus. And this leads to distortions that are kind of not as bad, and then normal methods for correcting for distortions would be able to correct for that. So, for example, field maps. Our second approach for this was that we used two different methods of localization. As I said, the localization is quite difficult, especially in functional MRI scans. So we used two different methods. One was an anatomically based method where we used an atlas that was published in 2018. And the second one was a functional localization method. So here the idea behind it is that in each participant, we would also give them a task that would reliably activate this brain region that we're interested in, but would be independent from our actual task that we try to study. And then we would use this, the functional activation from this region in each person to then functionally identify the brain region. So the way we did this was that we designed a task that was based on this finding that SNVTA neurons are sensitive to both reward and novelty. And we used the task that combined those two and then extracted the most active voxels, the top 100 active voxels that would show up in the contrast of reward novelty versus a non-reward and familiarity. So this is the task that we did in order to do this. So on the left-hand side, you can see that there are several pictures. The participants were, before the scan, memorizing these pictures, and they were counterbalanced across participants. So people looked at different pictures. And then during the actual task, they would, at the beginning of each trial, see a picture. And this one would be then either one that they had already familiarized or a new picture. And whether it was an old or new one would tell them whether they would be able to win money on the specific trial. So the actual task was then to press a button as fast as they could in response to a cue. And then if they would be fast enough, they would win money. Or on some trials, they would not be winning anything, even though they were fast enough. And these trials were signaled by pictures they had already memorized. So that allowed us to conflate novelty and reward in this cue. So we were able to make this contrast, which would then reliably activate this brain region and allow us to, in each person, functionally target the region of interest. So then once we had this region sort of identified, the way we then went about to study the signals of craving was that we used a cue-induced craving paradigm, which is a pretty standard and validated way how people have studied craving in drug studies. And what we did was that we asked participants to give us a list of their favorite foods, and then we created a picture set that would show them pictures of their favorite foods, as you can see below, with a short description to sort of increase the processing of the picture. And each picture was unique. So because the SNVTA is also sensitive to novelty, we tried to not conflate these dimensions. So each picture the participants saw in each scan was a unique one. So for control cues, we then used flowers, and we decided to do that because we wanted to have something that was sort of nice to look at, so somehow still also rewarding and also colorful, but it would not have a motivational sort of salience. So people would not crave flowers after fasting. So the way we then did the task was that we used a block design. Participants saw 3 pictures within each block, and then they rated how much they wanted food right now. And after the control cues, they would rate how much they like the cues to keep the motor response constant. So in terms of data analysis, I'm going to show you that now the approaches that we did and what we found using the food craving data. So in terms of univariate analyses, we used the 2 regions of interest that we had defined. This one region now, but in 2 different methods. So first, the anatomical localization and then the functional one. So on the left side, you can see an example for one participant how their activation looked like in response to this the task. So this was approximately how these activations look like. And from these voxels, we then tested how they would respond in the cue-induced craving task after fasting and after isolation. So here we showed that there was a stronger response in response to food cues after fasting in the anatomically selected region of interest and also in the functional region of interest, suggesting that the univariate response was showing modulation based on whether participants have been undergoing craving or not. And we also looked at the stratum, and for that we divided the stratum into 3 different subregions, and we corrected for multiple comparisons. So what we found here was that there was an interaction by region and session. So we saw that the nucleus accumbens was encoding or was responding stronger to pictures of food after fasting than after isolation, while the other regions did not show this effect. And this was in line with our whole brain analysis that also showed signals in the in the nucleus accumbens. So what we then looked at was whether the signals that we were seeing, like the changes in brain activation, whether that corresponded to a subjective experience of wanting food. And what we found was that there was a correlation between those two. So higher activity in the SNVTA went along with increased self-report of craving for food after fasting, suggesting that the activation in the SNVTA was directly related to this subjective experience of wanting food. And then in terms of multivariate analyses, so this was sort of just the first kind of check that we did, looking at whether there was any multivariate signal that we would be able to pick up on from the SNVTA, since most of the neurons there are, as I said in the beginning, dopaminergic, so they might respond in a very univariate way. So they might just have like this on-off firing. So it might be that the univariate patterns within this brain region are not very informative. So in order to test whether there was any signal that was sort of carrying meaning, we first did an analysis where we looked at whether we would be able to decode food from control cues in a session where participants were fasted. So here we used a linear support vector machine with 6-fold leave on out validation, which means that each fold was separated by each run. So we used 5 of the folds to train the classifier on the neural patterns in the SNVTA to differentiate between food and control cues. And then we used one left out fold to test the classifier by showing it a neural pattern, and then whether it would be able to classify this as a neural pattern associated with someone looking at pictures of food or pictures of flowers. And what we found was that the classification was significantly above chance. So the classifier was able to pick up on something that was represented within this brain region in a multivariate way, suggesting that there is a meaningful signal also in the multivariate analyses. So that suggested to us in conclusion that we were able to pick up on relevant signals representing craving in the midbrain using fMRI. So now we looked at how these signals would look like after fasting. And this is the task now shown for the actual full paradigm. So we also included social cues, and we used a similar method. We asked participants to give us a list of their favorite social activities, and we asked them to give us only a list of activities that they would always just do with others and never on their own. And we again created a data set of unique pictures. So as you can see here, these pictures are showing strangers engaging in certain interactions, and then we also included a brief description. And very obviously, this is not the only way that we could have done this. So one very sort of intuitive way that we also thought about initially would have been to show participants pictures of their actual friends or family members, and then measure the signals of craving. But however, we then decided to not do it that way because we thought that there would be a lot of sources of variance that we would not be able to control for if we did it that way. And one is, for example, the quality of the friendship. So we would not be able to really know like how close people actually were with those people. And then also the actual cues, we would not be able to control like the quality of the images and things like that. So we in the end decided to go this way, but it would certainly be interesting to test it also with using actual pictures of friends. So now just to summarize our main hypotheses for the social isolation, which were all pre-registered. So we predicted that the SNVTA would be modulated by isolation, and that this activity would correlate with ratings of social craving, and that the patterns in response to food fasting and social isolation would share a common neural signature. So first, we looked at the univariate signals to answer these hypotheses. So again, we looked at the two different ways how we were localizing the SNVTA. And what we found here was that in our anatomical localization, even though there was a trend, we did not find a significant effect of isolation on brain responses in a response to social cues. However, for the functional ROI, we did find it, and that was also significant if we removed the outlier that you can see there. So when we used the voxels that were individually selected, we saw that the SNVTA was responding stronger to social cues after isolation than after fasting. And then we looked at the striatum, and here what we found was that there was again a region by session interaction, but here we found that it was a different region within the striatum that was coding for craving of food, and that was the caudate. So we found for the caudate that it was responding stronger to social cues after isolation compared to fasting, but not for the nucleus accumbens as we saw for the food fasting data. And that was also in line with the whole brain analysis that showed a very similar result. So then we looked at the correlation with subjective craving. So here what we found again was that there was a correlation between how people rated how much they wanted to engage in social contact right now and how strongly their SNVTA was activated. So those people who were more social interaction showed stronger activation, suggesting again that there was a direct mapping between these brain signals and the subjective wanting of social contact. So then as a final test, we looked at the multivariate pattern. And the way we did it for this test was that we used a cross-classification method, again, using a linear support vector machine. And what I mean by cross-classification is that we used one session to train the data on, which was the fasting session. So we trained the classifier again to classify food from control cues after participants were fasted. But then we tested the classifier on another session. So we did not leave out any specific runs. We just used all of the runs from each session. So first of all, we tested from the isolation session whether the classifier would again be able to classify food from control cues, suggesting that there was a generalization for the stimulus, so seeing a food cue. But the more interesting test was then to look at whether this classifier would also be able to classify social from control cues. So and this test is crucial because for these patterns, so the classifier had never sort of seen the neural patterns associated with a lonely person looking at social cues. It was only trained on these patterns of hungry people looking at food cues. So if we would be able to sort of cross-classify across sessions and across stimuli, there would be, this would suggest that there is a shared underlying neural signature between this sort of craving for food and the craving for social contact. And then as a comparison condition, we also did the same thing for the baseline condition. So what we found for this test was that for the food cues, we were able to cross-classify food from control cues in all sessions, suggesting that there was a stable representation of food versus control cues across sessions. For the social cues, what we found was, more interestingly, that we were able to cross-classify the cues in the isolation session, but not in the baseline session. So below you can see the confusion matrices, and you can see that so apparently there was information there in the social session that the classifier was able to pick up on, and then allowing the classifier to above chance classify the pictures, but in the baseline session, it was not there anymore. So this suggests that there is something about this state of deprivation that is sort of stable across these two sessions. And to look at this finding sort of more closely, we then also did representational similarity analysis. So that allowed us to quantify the representational distance between conditions. So we used the distance measure of 1 minus correlation and between these 3 sessions, and I'll just walk you through what we mean by that. So food craved means someone who's looking at, or the neural patterns of someone who's looking at food pictures after being fasted, and social craved would be someone who's looking at social pictures after being isolated. So the 2 conditions share in common that there is a similar motivational state. In both states, people crave something, but the cue that they see is a different one. And here, in this other test that we did, we have food craved versus food non-craved, which means that this is the response to food cues after being isolated, where we knew that people had eaten a lot because we gave them food. So they were not hungry, but they were looking at cues of or pictures of food in both conditions. So this is the stimulus that is constant. And the third condition is the maximally dissimilar condition. So here we have both the cues and the motivational state are different. So this was sort of our ceiling. So this should be like the value that shows the strongest dissimilarity. And then we would compare the other 2 conditions to this one. And then testing whether it would be rather the motivational state or the stimulus that would be more similar than the maximal dissimilar one. And what we found was that it was actually the motivational state that was the most similar. So in this graph, you can see the representational dissimilarity. So a larger bar means that it's more dissimilar, and the smaller one that it's more similar. And there was a significant difference between the motivational state and the ceiling. And the direct comparison between motivational state and stimulus was trend significant. So this suggests that the neural patterns between someone who has been fasted and looks at pictures of food and the neural patterns of the same person if they have been isolated and looking at pictures of social cues are more similar than if the same person is looking at the same kinds of cues but is in a different motivational state. So this suggests that the SNVTA seems to code very strongly for this for the motivational state, and that the craving between those 2 has something very similar within these neural patterns. So to sum up, this study, what we found was that, first of all, we could sort of confirm our first hypothesis. So we did find that SNVTA activity was modulated by isolation, but only in the midbrain voxels that were most sensitive to reward. So these that we selected using our functional localization method. What we also found was that SNVTA activity correlated with ratings of social craving, and that that the patterns between food fasting and social isolation share a common neural signature. So the implications for this are that, first of all, the midbrain encodes social craving, which is consistent with the results that were shown in the mouse model, and that social craving signals are very similar to those of craving food. And this suggests or supports this so-called social homeostasis model that suggests that social contact or our motivation for social contact operates in a very similar way like other motivated behaviors. So there's sort of a set point that we have where we try to reach that, which is the homeostasis. And whenever we detect like a deviation from that, we are motivated to engage in behaviors that would let this set state go back to normal. So if you compare this to food, so someone would not want to be hungry, and whenever they're hungry, they need nutrition. So a hunger would motivate us to go out and eat. And the same seems to be sort of true for our social contact and why we experience loneliness. So it seems to be something like a motivational drive to engage with others. So one other thing that our results also suggested is that our experimental paradigm was successful as an experimental manipulation to induce feelings of loneliness and isolation in people. So 10 hours of isolation led to increased feelings of loneliness and social craving, and altered neural responses to social cues in motivation brain areas, suggesting that we can use this paradigm to study sort of in an experimental setting how isolation and loneliness affect our behavior in our brain. So now I will go on to talk a bit about the sort of similarity or dissimilarity between acute and chronic isolation. So in our study, what we found was that isolation led to changes in dopaminergic midbrain neurons, or voxels in this case, which then also changed social approach behavior. However, this is true for acute isolation. Studies on chronic isolation, however, in the literature have shown that chronic isolation via a mechanism that we don't know actually leads to social withdrawal. So loneliness is associated with lower social approach motivation. So is this a potential contradiction between our studies and studies on loneliness? So we kind of then also tried to look into this. And the way we did it was that we had collected data or self-report data of each participant about on their experience of chronic loneliness in their everyday life before they joined our study. So we were able to look at how chronic loneliness was modulating this relationship between how people responded to social isolation, both on the level of the brain and on the level of self-report. So here I'm just showing the measures that we used for these analyses. And what we found was then that there was an interaction. And indeed, if people had reported higher loneliness before joining the study, they actually responded less to the experimental manipulation. So those people showed lower activity in the SMVTA in response to social cues after being isolated. And what they also showed was a lower self-reported craving for social contact. Which I'm not showing here, but it was a similar relationship. So this suggests that loneliness somehow either that it makes people want less contact, or perhaps people do not, or maybe they are not that sensitive to this experimental manipulation. It's not quite clear, but in any case, there is an interaction. So one potential explanation could be that that acute deprivation of social needs sort of engages regulatory processes like social craving and hypervigilance to social cues. And when people, however, experience this deprivation state over long term, it might be that the sustained recruitment of these regulatory processes leads to their breakdown, and in a way then to the shutdown, which is sort of maybe a similar thing as is going on in acute versus chronic stress. So we know that acute stress is something that is a very natural, normal, and healthy response to being sort of like encountering any form of challenge. However, if we experience stress over prolonged durations of time, we know that this leads to pathology, to these systems sort of not working properly anymore, and people developing stress-related disorders. So it could be that there is something going on there, that the long-term exposure to isolation sort of changes something in these processes. However, we don't, again, quite know, because also in our study, the causality is not quite clear, as it could also be very well that people, even before ever becoming lonely, actually already show decreased activation or responsiveness of these brain regions, and maybe just care less about kind of experiencing loneliness and things like that. So I think this is kind of an interesting avenue for future research to look more into this interaction, how people who already are lonely sort of respond to kind of deficiency of social contact. So now I would go on to talk about the next steps in my research and what I'm doing currently, but I'll just do a quick break and ask if anyone has any questions about this first part. I mean, we can also discuss them at the end, but maybe someone has a urgent question.
Anyone in the audience can either raise his hand or write something in the chat or Q&A boxes.
If there are no questions, I can just go on, but yeah.
Nothing yet.
Oh, okay, good. So then I'll just continue with what the next steps are. So one thing that I thought was a very fascinating but also sort of alarming finding was that there was a study in 2019 that was done in a really large sample in the UK. So these were more than 50,000 people joining. And what they found was that they were looking at loneliness in different age groups. And what they found was that it was actually young people who reported the highest levels of loneliness. So even though most people think intuitively that it's more the older people or people, older people who are the most lonely, the study and other studies actually as well have shown that it's actually rather these adolescents and young adults who experience the highest level in loneliness. And this is not something that has been always the case. So in 1970, this age group reported about 11 to 17%.
Livia, can I interrupt you for one
Yes,
second? So we just have one question from the audience. Bruce Jenkins, can you please unmute?
Oh yeah, I'm sorry. I had raised my hand, but I guess I didn't get noticed. But this goes to the prior part of the talk.
Okay.
Did you actually look at kind of the networks that were involved in the social isolation versus food? Because, for instance, some studies have shown differences in the networks and the actual basal ganglia striatal localizations of reward signals for and reward circuitry for food versus, for instance, drugs like cocaine. And there's a whole panoply of brain networks that one can investigate, and it seems like you were very hyper focused on just the basal ganglia and the nigra.
Yeah, so we did that as well. So our pre-registered and our main hypotheses were concerning the substantia nigra and the striatum, but we then also, as you said, there is a kind of craving circuit, and we looked at those regions as well. And in those regions, we then found that there was actually a dissociation. So we saw that there was a representation of social craving in the ventromedial prefrontal cortex, but food fasting was represented mostly in the ACC, I think. I don't know. I'm sorry. I will have to check the paper again. But anyway, we looked at these brain regions and we did not find that they all responded the same way. So this suggested to us that it looks like at the sort of most basic level, those social craving and food craving are very similar. But once we sort of go further into the brain, and we also saw that already at the level of the striatum, the signal seems to be dissociated. And I guess that makes sense because they have to be somewhere like different on some level. So it suggests that there is like a similarity, but also dissimilarity in how social craving is represented across the brain. Does that answer your question?
Yeah, that's a good. The other thing I was interested in, just if I can ask one quick question more, is you're getting a lot of the signal changes you're seeing in the nigra were actually negative. If you looked at the percent signal change, a lot of them were negative. And it kind of begs the question of, are you trying to interpret the fMRI signal changes as either, activation of that network or inhibition of that network?
Right.
Because, it doesn't necessarily have to be activation or inhibition if you have a positive or negative signal, because there are, different receptors that control either negative or positive hemodynamic changes, in the nigra. So the question is, I'm just curious as to what you think about those findings, or if they actually mean anything as far as which subjects showed negative signal changes and which showed positive.
Mm-hmm. So what we saw on average was that when we made the contrast between responses to the social cues or food cues versus the control cues, on average the signal was positive. So we did see the activation when we just looked at the raw signals, so the betas. But when we did the contrast, we saw that the signal was positive. But anyway, it was more positive compared to the control cues. So I think it's hard to interpret the raw signals in itself, mostly also because if you think of the analysis, how you implement a GLM, you would have like this sort of implicit baseline that you would compare to the whole brain. So then if you were looking only at this at the region, which is the SNVTA, it's sort of hard to first of all compare that, and the second thing is that in general, what does baseline activity mean? So there are papers showing that you should mostly interpret a contrast, which makes it like the most straightforward way of analyzing data. And if we look at the difference between the two, we do see that the activation is higher in response to the either food or social cues after the deprivation. I hope that answers the question.
Well, somewhat, but okay, thank you.
Okay.
Thanks.
Okay, thanks. Okay, so I'll continue with this part now. All right, so I was speaking about the loneliness in this age group and that it increases over the past years. So in the study that was done in 2019, we saw that it was 40% of other people in this age group who reported to be lonely. And this is problematic because we see that many mental health disorders have their onset in adolescence. So adolescence has been most recently defined as the age range between 10 to 24 years. And we see that a lot of mental health problems start in this age range. So it's kind of important to look more into this connection between isolation and possibly the effects of it if this age group feels the most lonely and is also the one that shows these onsets of mental health disorders. And I'm not sure if I should go on. So it's like 10 more minutes and I can skip some slides and we can just do the discussion or I'm not sure how.
It's fine. You can carry on if you want.
Okay.
There's no time.
Okay, cool. Yeah, so the definition of adolescence has been most recently put to 10 to 24 years, and this is mostly based on studies looking at brain development that have shown that brain development actually continues up until the age of 24. So I'm just showing here like a summary of different studies across different countries, and we see that matter volume increases annually during adolescence by 1%, and that goes up until around the age of 24, when it then plateaus. And we also see that gray matter, which is decreasing throughout development, and that this development continues again up until the age of 24 approximately. So gray matter decreases by around 1.5% annually during adolescence. And the underlying sort of macrostructural changes that are thought to underlie this is, first of all, for the increase in white matter, the assumption is that it's myelination and the growth of axons that lead to this increase in white matter throughout development, and for the decrease that it's synaptic pruning. So on the right side, you can see dendrites and their dendritic spines, and you can see that in the age of between 1 month and 2.5 years, there's an overexpression of these spikes, but then there is a pruning away of those for the ones that are not used. So this is thought to be an important developmental change that happens until quite late into our development. So one thing that I'm interested in is to look at how isolation affects the developing brain, and specifically in adolescence. And I'm most interested in adolescence because it's a time that is characterized by a strong sensitivity to social influence and social cues. So adolescents are very interested in other people. They are very sensitive to positive and negative social cues. And they also show this reorientation to their peers. So this might suggest that if they are experiencing isolation, this might have strong effects. And indeed, there are animal models that look into that. Many animals, actually most animals, undergo very similar developmental stages like humans do. So for example, one animal that is studied often in these studies is the rat or the mouse, and they both undergo like similar developmental stages. So, and they also have a sort of a phase in their life that is very similar to human adolescence, that is also characterized by this sort of reorientation to peers and this increased insensitivity to rewards. So what people have looked at was how adolescent animals respond to either short-term isolation, so that has been characterized between 24 hours and 1 week, or long-term isolation, which is over a week. So to summarize these studies, what has been shown is that short-term isolation in general seems to be experienced as a stressor, so it leads to increased activity in the HPA axis, and it increases dopamine metabolism, so there is an increased sensitivity to rewards, and specifically social rewards. Importantly, reintroduction of these animals into social contact reverses most of these effects. So for long-term isolation, the changes are similar but kind of more pronounced. So what has been shown here is that there is a dysregulation of activity in brain reward systems, which then leads to a strong reward seeking in these animals, but a decrease in reward learning. So they somehow strongly respond to rewards, but once the associations are established, they are really bad at shifting flexibly. So they show a very repetitive behavior, and they are also more prone to develop addictions or addictions if given the chance of given the access to drugs or alcohol or to overeat. And there's also changes that have been observed very consistently in serotonin activity, which goes along with increased responsiveness to stressors and in general this increased anxiety that these animals show. In addition, what has been also found in these studies that at the level of structural brain development, there seems to be also an effect. So they found that there is a reduced synaptic pruning if animals undergo adolescence in isolation, and that has effects on executive functions in these animals. And importantly, these changes are not reversed after reintroduction to, into social contact, suggesting that these are more long-term, and they don't change anymore. So one question that I'm interested in is how can we translate potentially these findings to humans, and do humans show similar effects of isolation? So what are the effects of motivation and cognition on adolescents? So one way to potentially study this is again using short-term isolation as a model to sort of look at how adolescents respond to being alone for a brief duration, whether they do show increases in reward sensitivity or decreases in flexibility of learning. So one study that I'm currently, we actually were supposed to start scanning already, but are not scanning now due to lockdown. But the study that we're doing or trying to do right now is that we look at adolescents in this age range that has been shown to be for those people who are the most lonely. And then we use a similar setup as I did in my previous study, but with a shorter duration of isolation since these participants are younger. And it will be variable, so they would not be able to directly tell how long the duration will be, but not longer than 4 hours. And then we would test how isolation would affect things like effort-based decision-making, flexible learning, cognitive control, and risk-taking. And one other thing that I'm very interested in is how virtual social interactions interact in these effects. So if people are isolated but have access to social media, does that remediate the effects of isolation, or does it maybe even increase them? So there are, in the present literature, there are sort of contradictory findings showing either that social media use can increase loneliness or increase social connection. So it's not quite clear. And it has been suggested that it strongly depends on how people use social media, so whether they use it actively or passively, but there are no real studies, or very few studies, that look directly at causality, like how does certain engagement with social media affect our feelings of loneliness. So these are the project that I'll do next, and at the end now, so I'll want to thank all the people I worked with and the funders and you for your attention. Thanks.
Dr. Tomova, thank you so much for this great talk. We now move to the Q&A phase. So again, everyone, you can address any question you have using the Q&A box, or raise your hand and we'll try to monitor it better this time. On the meanwhile, I'll just ask if you have any specific plans to look at COVID-related isolated patients, either the regular population or just or patients or elderly.
Yeah, that's that's definitely a very interesting question. So we're currently doing a project where we're looking at effects of like COVID-related social distancing on feelings of loneliness and mental health, but we're not doing this in an fMRI study, but in an online study. So our goal here is to look at how people, like in what kinds of behaviors people engage in, and whether these sort of mediate how people then experience loneliness and whether that affects their mental health. So the way we want to do this is to use a tracking method where we would see how people actually use their phones. So how they would engage in social media and how much time they would spend on it, and then see whether there would be a relationship between these types of usage and how people then whether they feel lonely due to social distancing or not. Because as I said initially, not everyone who is alone physically is actually necessarily experiencing loneliness. So it's sort of unclear, like, who are the people who then develop loneliness and why is this the case? So we're trying to look more into this.
Great. Another question, I'm not sure if it's relevant or if you had enough data or participant, but did you consider using another classifier rather than support vector machine, maybe an artificial neural net? Is it relevant?
I think that's an interesting question. I think in theory, of course, we could have tried different sorts of classifiers. I mean, one thing against the neural networks is that it's really hard to tell how they, like, what kinds of data they or input they picked up on. So it would be hard to then, I mean, since our goal was not to just be able to classify one condition from the other, but actually be able to tell, well, what does it tell us about the cognitive process? The SVM classification allowed us to be more, or to have more control in that, because we would be the ones labeling the features, we would be the ones knowing what we trained on and what the classifier was training on. So it was sort of, it seemed like a more straightforward approach. I think it would be interesting to try to train a neural network, but again, it would be hard then to tell what was the actual activation it picked up on, and yeah, so.
Thank you. Just a sec, I think someone raised his hand. Bruce, you can unmute.
Unless there's someone else who wants to ask a question, I don't want to monopolize questions.
You're the next one.
Okay, actually, just related to the last question, how did you select the feature variables that you used in the classifier?
Right, so we selected the beta values from each condition. So from each session, the beta value in response to or associated with food pictures. So for example, after craving. So these were the ones that we selected.
And what thing I was curious about is, have you looked into the relationships between the social isolation and depression? Because it seems there's a good deal of overlap between those two conditions, and certainly, for instance, for diseases later in life like Alzheimer's, there's definitely risk factors associated with both social isolation and depression. So I was just curious what relationships may or may not exist between those two, or have you looked at?
Yeah, I think that's a really interesting question. And I mean, obviously those two are related. Like in either direction, people who are lonely often become depressed. And we know that people who are depressed tend to self-isolate themselves often. So one thing that, I mean, we did not directly look at the relationship, mostly because we did not have data from our participants on like depression scores and things like that, even though that was something in hindsight that we would have, like, we thought about this when we analyzed the data, and we did think that we should have collected this. Anyway, one thing that I wanted to say is that in addition to what we found in this interaction between chronic loneliness and the acute responses to social isolation, where loneliness showed like sort of this, people who are more lonely had like these dampened responses, we actually also found this association to the food fasting condition. So people who were chronically more lonely also showed lower signals in the SNVTA in response to food cues after fasting, and also self-reported wanting less food after being fasted. So that suggests that maybe there is something more broad going on, that there's in general something going on with the motivation system, that it's in some way dampened if people experience chronic loneliness in comparison to others. So I think that's a really interesting finding, and I'm really keen on sort of following up on this and looking at this relationship between, in general, motivated behavior, processing of motivation, and loneliness, and how that interacts. But again, from our study, we cannot tell the direction. So it could again be that loneliness is causing these effects, or that people who already have maybe some like decreased responsiveness of their motivation system are the ones that end up becoming more lonely, because maybe they have less drive to interact with others. So it's really hard to tell at this stage. But I definitely agree. It's a super interesting question whether and how these two are related.
Thank you. Nice talk. Thank you.
Okay, so again, Dr. Tomova, thank you so much for this great talk. We hope things will look better in the UK and worldwide than over here, and we could have you in person soon.
Yeah, thanks for having me. And yeah, that would be nice.
Thank you very much. Thank you, everyone. Goodbye.